pheno red tubes are going to have the Durham tube inside. So one of the first things we want to do is we want to look to see if we see a gas bubble. And the reason for that is if we see a gas bubble, even if we don't see a color change, that indicates that the carb is being broken down. And you might be going, well, wait a minute, how can I get gas without an acid? Because sometimes our organisms have enzymes, and as soon as they make that acid, they're going to convert it to something else. Alright, so the acid's not going to be able to build up high enough to cause a color change. So that can happen. So in your book, it asks you to record if you see a gas bubble or not. So when you're looking at your media, the Durham tubes are sticking up. I'm looking to see if there's a gas bubble at the top or if there's an obvious gap in my media. Okay, so that's where we're going to start. The next thing I want to know is, did my organism turn a different color? So remember, what color did they all start out? Red. Yeah, pink. There was so for glucose it was red, is what we started with. And then for lactose and sucrose, unfortunately, they were kind of pinkish red because we were having some pH issues. So they were pinkish red. So let's start with our glucose. Did any of our organisms turn the media a yellow color? Yes. Good. So which organism turned the media bright yellow? E. coli. Good. So what that means then is E. coli is going to be the organism that is positive for breaking glucose down. Now you might be looking at this and going, well, wait a minute. I thought Pseudomonas aeruginosa should be able to eat glucose too. Ooh. Let's talk about Pseudomonas aeruginosa. Pseudomonas aeruginosa was mentioned in lecture, and we said it was a throwback. He's a flip phone user. Which pathway does he use? The ED pathway. Good. So in the ED pathway, I'm making some pyruvic acid, but I'm not making a lot of energy. So with this guy, if I'm only giving him glucose, and he's in this broth, there's not a lot of nutrients there, he's not going to be able to survive for very long. He needs additional nutrients to support his lame pathway, even though he's insistent on using that flip phone. You know, we just wish he would update, but he won't. So he can eat glucose a little, but not enough to change the acid content. So not enough for us to see a color change. So we would report him as being negative, because we're not seeing a reaction change. All right? Does that make sense? So we're looking for acidity to build up. Yeah.
So he's negative for the acid and gas, but is there still some fermentation going on?
We're going to say negative because we're looking for either yellow or red.
Okay.
It is a very clear, clear test. I want yellow or I want red. All right, let's go to the lactose test. We had E. aerogenes and Proteus vulgaris, correct? Okay. So which organism is showing a yellow coloration? Good, I heard it. Aerogenes. Yeah, E. aerogenes and Pterococcus aerogenes should be showing a nice yellow color. Now you might notice that Proteus looks a little orangey. That's okay if he looks a little orangey, he's still not a pure yellow. We are looking for red or yellow. So what that's telling us is he's making a little acid, but not enough to give me a strong positive. Alright? So E. aerogenes should be positive, Proteus vulgaris should be negative. We go to the last one, sucrose. I had E. coli and Proteus vulgaris there. We're giving Proteus another chance. Which one of these turned yellow this time? Proteus, good. So Proteus vulgaris is going to be positive. So the reason that this is important, if I was to do a gram stain of E. coli, of Proteus, and of Pseudomonas, they all look identical. They're gram-negative rods, they're all teeny tiny, they all look the same. So if I was to stain these, I wouldn't be able to tell a difference between them. So using this type of media is a way that I can quickly determine differences. Do y'all remember that book I showed you at the beginning of lab called the Bergey's Manual, and I flipped it open and there were all those tables inside? Those tables are showing when we're having sugar reactions. So I can set these broths up really fast. I come in the next day, and if I see glucose positive, sucrose negative, that's a fingerprint for E. coli. Make sense? So this is how we're using them. We're using them to look for identification characteristics. What kind of sugars are they eating? What's their preference to survive? Because they're all going to look the same. This is going to help wanting to key them out. All right? Everybody with me on that? Okay, all right, we're gonna go on to the next one. The next one is Simmons citrate. So for Simmons citrate, remember, this is one of our few chemically defined media, okay? So chemically defined means what about the media? If something's chemically defined, what does that mean? I know all the ingredients. Yeah, I know every ingredient. Do we use chemically defined media very often?
No.
No. Remember, it's expensive. It's a pain to set that up because it's a lot of work. But it's important when I need to know every single ingredient. Okay? So in my Simmons citrate, the only carbon source was citrate. Okay? So citrate's a big, complex carb. My organism has to be able to produce enzymes to break these carbs apart and then rebuild them in whatever structure it wants. So that's the whole point, is I'm using an enzyme and I'm taking my Lego structure apart and I'm rebuilding it into something else. Okay? If I don't have this enzyme, citrase, then I can't do that restructuring. Now, if I'm releasing carbons, then that means, remember, how do carbons get released? What do they release as? CO2s. So if I'm releasing carbons, and if I'm maybe not using them all, I'm releasing CO2. And CO2 is going to be basic, which is going to raise my pH. Okay, so I'm going to get a color shift. So looking at your organism, the negative should be a green color, the positive should be a really pretty blue. That means that I got a CO2 change. I went more basic. Okay, so who is presenting as a pretty blue? E. Erogenes. Good. E. Erogenes should be positive. E. coli should be negative. So questions that I would ask on a lab exam. You set up Simmons citrate. You come in the next day. You see an organism that has turned blue. Which organism was positive for a blue color? Well, first of all, I could ask you, is blue positive or negative? And then I could ask you, which organism do we see in the lab that caused a positive reaction? And then I could ask you, which organism was negative? So these are the kind of things you want to study. And then I could say, what does that mean? What's the whole point of this test? And then I could ask you, is it chemically defined or is it complex? So these are the kind of things you want to make sure you're understanding the results. Okay, so understand the results. I would start by memorizing the organisms and their reactions, and then dive deep into understanding what's happening. All right? Very good. Yes?
Real quick, this case, increased in carbon dioxide, not being reduced to carbonic acid.
In this case, no, because they're using it to build other carbon structures.
No.
So it's a very brief shift. It's enough to get a basic shift, but it's not staying for long because I'm immediately putting it into a new Lego structure.
Okay.
Yeah, that's a good question. Okay, MRVP. We're going to read that guy out next time. So I want you to take your MRVPs, put him to the side. He's going to go back in the incubator with your proteins today. All right? Do not throw them away. Make sure you can put them on your racks that have your stocks that we're going to set up with today if you want to make sure we don't lose them. But he's going to go back in the incubator with all of your new tubes today. The reason for that, the MR test is usually very quick and easy, and we get a really quick response. The Voges Crossbow, the VP, sometimes it needs an extra few days, so I'm just going to wait and let it cook a little longer. I need the bacteria to get up a little higher, all right? And I just want to make sure, so we're going to cook them, and then we'll look at them next time. So this one you can skip for today. We'll be reading them out next time. Okay, last but not least, OF media. So our OF media, what color did it start? Started green. Started green. So if I'm oxidative, that means that I'm going to be able to turn a yellow color, and in this case I'm looking for a yellow color when oxygen's present. Okay, I know oxidation reduction doesn't really have anything to do with oxygen. In this case we're using oxidation, losing electrons, meaning the glucose and sugars are being broken down, and we're getting a pH shift. So if I turn yellow with oxygen, then that means that I'm positive for an oxidative reaction. Okay, oxidative, not oxidation, but oxidative. Okay, which organism organisms turn yellow? Okay, did our Pseudomonas turn yellow at all? He should. Let's see. Yeah, he's yellow. So see at the top how he turned yellow? So Pseudomonas is also yellow as long as there's oxygen present. Do you see how he doesn't turn yellow really deep? Yeah, he's just at the top. That makes sense, right? What did we learn about Pseudomonas in our lecture? What kind of atmosphere does he prefer? Yeah, he's aerobic. He's a obligate aerobe, just like us. All right. So Pseudomonas is going to turn yellow, and so is E. coli. Now E. coli turns yellow throughout. Pseudomonas turns yellow at the top, but they are both going to be positive. All right. How about fermentative? So this is where I added oil to the media. Which one of my organisms was able to turn yellow when there was no oxygen present? E. coli. And again, hopefully it makes sense, right? What kind of environment does E. coli prefer? He's a facultative anaerobe. So he likes oxygen better. When oxygen's present, he can get 38 ATP. But if I take oxygen away, he can make it. All right? He's a facultative anaerobe. He can live on those 2 ATP. We'll figure it out. Just have a little cookie. And that makes sense. Where does E. coli live in our bodies? In our guts. Are our guts aerobic or anaerobic? Anaerobic. Anaerobic. Yeah, I hope so. Yeah, they're anaerobic, okay? So they're going to be anaerobic. If we got air bubbles in there, we're not going to be feeling good, all right? So yeah, they're anaerobic. So E. coli survives in our bodies, and then if we put E. coli on a plate, he lives in our incubator, so he can survive either way, all right? So E. coli can go either way because he's a facultative anaerobe. Okay, does that make sense? Okay, good. Make sure you do not lose your MRVPs. We are reading them out next time, so don't toss them. Make sure you set those to the side in a safe spot. Do not lose them, because we will be reading them out next time. Okay, today's lab is going to be setting up protein catabolism tubes. So just like what we did in lab 12, we were looking at organisms that were able to break down carbohydrates. In this case, we're going to be looking at organisms that are able to break down proteins. Now, depending on the enzymes that I have, I will break my proteins down in different ways, all right? So if I put a break here at this carbon and I break off an amino group, that means that I'm going to be producing an enzyme called deaminase. Deaminase. I'm removing the amine group. And if I remove an amine group, that means that my pH is going to drop, okay? Because if I release a nitrogen with 2 hydrogens, it's more negative, it's going to drop my pH. Okay, so I'm going to get a little bit of an acid shift. Now, other times, I might have an organism that produces an enzyme that breaks here. And if I break here, I'm releasing the carboxyl group. What did we learn about our CO2s over there? What did they do to my media, my pH? They went up, yeah. The same is true when I release a carboxyl group. So if I release a carboxyl group, my pH is going to increase, okay? We call that decarboxylase. So as an organism, when I'm living in the environment, I will either produce deaminase or decarboxylase, depending on what kind of nutrients are available and what enzymes I have, okay? So when we start talking about our proteins, we're going to learn that some of the media looks for deaminase and some of the media looks for decarboxylase, okay? And we'll be getting into depth on those next time. So today you're going to be setting up only 2 of the media types because we don't have the 3rd one. So the ones that you're going to be setting up today is the phenylalanine slant. So you're going to be setting that up with E. coli, and then your lab partner can set that up with Proteus vulgaris. So you're going to set up the slant, and that's going to be how we usually do, where we just are going to streak along the surface. So pretty quick and easy. And then my next one that I'm going to set up is the MIOD. We do not have lysine decarboxylase available, so this one you can just kind of cross off. We don't have that one. The MIOD, you're going to set up with E. coli and Klebsiella. The way you're going to do that is with a about 3/4 of the way down, and then pull out. Now, the MIO is going to actually be a really cool test for us because it's a 3-for-1. This test is going to be checking for motility. It's going to be checking for something called indole, which we'll talk about next time, and something called ornithine. So this test, microbiologists love it because we get 3 results in a single tube. That works really well for us. So imagine like over here, we did our glucose, lactose, and sucrose. We had to do 3 different tubes. But over here, in just one tube, I get 3 different test results. That's a lot of bang for your buck. So we all like these. So the MIO test that we're going to set up today, we'll read it out next time and we'll talk about how these are going to relate to our results. All right, we'll go into more detail next time. Again, we don't have lysine decarboxylase broth. I'll try to find an image that we can look at so that you can at least see it next time. The result is actually very similar to one of the tests here. So it was not a bad one to lose. We'll talk about how it relates to this media. Okay, does everybody have the game plan? When you're done, all of your used tubes except for the MRVP go to the side of the room. All of the new tubes that you're setting up with the MRVP are going into the incubator. All right? Also, I was told that the E. coli didn't grow very well. I'm not sure why. So I do have E. coli up here on a plate. So when you're ready to set up your E. coli, you can go ahead and come up here, and I'll put the Bunsen burner on, and you can set up your E. coli up front. Okay, so then that way everybody can kind of come up and set up here. Or if you take it back, you can make sure you share with your lab partners, whatever you guys want. You guys want to do that? Do you want to take it back on your bench? I'm fine with that. It's just I don't have 6 for each bench, so I only have like 4, so it's going to be